Hello, my name is Chelsea Price and today I will be presenting the article Field-Based Evaluation of Scat DNA Methods to Estimate Population Abundance of Spotted Tailed Quoll, Dasyurus maculatus, a rare Australian marsupial, published in Wildlife Research 2009. This study aimed to critically assess the reliability of the non-invasive method to estimate population abundance of the spotted tailed quoll. The most effective method to survey quoll populations is through live trapping using cage traps. This method is not ideal in some situations as the quoll range is large and largely rugged with limited vehicle access.
The spotted-tailed quoll, Dasyurus maculatus, is a medium-sized carnivorous marsupial of eastern Australia and is listed as endangered. The decline of this species is unknown, as these methods of estimating the population abundance are difficult to conduct.
This study was conducted by the use of traps over a 7,000 hectare area of Bataboo Wilderness Area, located within the Kosciuszko National Park in southeastern New South Wales, Australia.
Figure 1 shows the location of the study area and the size. The squares on Figure 1 show the locations of the scat collection and the live capture cage trapping sites that were conducted between April and July 2005.
Trapped individuals are permanently marked for unique identification with a passive integrated transponder and weighed to estimate age. An ear biopsy was taken from each individual to use as a reference for comparison of the scat DNA. Quoll scats were collected from 20 trapping sites where active latrine sites were present, and another 10 scats were collected from sites where traps had not been set. 16 other sites were not included as the latrines were not monitored or were not present at the trapping sites. DNA identifying methods were used to identify individual quolls for population abundance estimates. The population estimates were developed by the comparison of the data from the trapping and the scat collection. During May and July 2005, a total of 22 individual quolls were captured over 1,563 nights and on 109 occasions. These included 6 females, 15 males and 1 intersexual individual. During April to July 2005, 262 scat samples were collected. 18 individual scats were identified as the trapping individuals. All 6 females, the intersexual individual, and 11 of the 15 males were identified. 75 latrine sites were monitored for the scats over the duration of the study.
As shown in Table 5, the scat complement and May to June scat datasets were similar to the estimates of the trapping data. The mean estimate derived for the scat dataset was within 1 to 6 individuals and 1 to 4 individuals for the trapping dataset and May to June scat dataset.
This study highlighted the efficiency of estimating abundance of populations of the endangered spotted tailed quoll using collected scat DNA. This evaluation provides meaningful estimates of population abundance. The results demonstrated that DNA surveying can identify animals that had not been trapped along with the ones that were. The results confirm that scat samples can be used to obtain individual profiles that can estimate population abundance. In conclusion, this study provided the very first evaluation of population abundance estimates by using scat DNA collected from free-ranging spotted-tailed quolls. The study suggested that with DNA technology improvements to allow the use of older scats, the non-invasive method would be a practical alternative to trapping in isolated and inaccessible areas. The study proved that scat DNA sampling was a feasible method to detect individuals and estimate population abundance.